Welcome to our webinar today, everyone. We thank you for your active participation. I'm Dr. Tegyu from WUMBI, the moderator for this webinar. This webinar is co-organized by WUMBI and Codon Genomics. And in this seminar, we will discuss about molecular medicine in the era of digitization and data analytics. First, we would like to invite the director of WUMBI, Associate Professor Dr. Azian, to give the opening speech. Please welcome Associate Professor Dr. Azian.
Thank you, Dr. Tekyu. Assalamualaikum and good afternoon, everyone. First of all, I would like to welcome all the speakers and participants to the Molecular Medicine in the Era of Digitalization and Data Analytics webinar. This webinar is one of WUMBI initiative to strengthen collaborative work with the industry. With this initiative, we hope to increase our research and learning quality, also to help the researchers and lecturers to produce high impact research output in future. Big data analysis has the potential to reduce costs of treatment, predict outbreaks of epidemic, avoid preventable diseases, and improve the quality of life in general. In this webinar, the expert in this field will highlight several important topics on big data analysis from the next generation sequencing and genome-wide association study in diseases that are very prevalent in the Malaysian healthcare system. I thank the speakers once again from WUMBI, Dr. Nurul Shakima Abdul Muttalib, Dr. Nur Aidatul Akmal Abdullah, Dr. Muhammad Irfan Abdul Jalal, and speakers from Codon Genomics, Su Bun Chua, Ching Gilau, and Dr. Wei Kang Lee. I hope all the participants will benefit from this sharing session, and we welcome any future collaborations. Thank you, Doctor. Thank you.
Associate Professor Dr. Azian for your kind support for this seminar. And now, dear participants, thank you for being with us. Before the speakers start their presentations, I would like to share some important information with you all. So today we have a very packed program whereby we have 6 speakers, 3 each from WUMBI and Codon Genomics, will share with you their latest research. So in our program, we will allow all 6 speakers to finish their respective presentation first. And finally, we will open the session for Q&A, which should last about 30 minutes. So, but due to everyone's available internet bandwidth, all participants are encouraged to type out your question using the Q&A features in the Zoom chat box or comment in our Facebook broadcast. We will compile your question and redirect them to the speaker for them to answer. So in your question, please specify first your name, then the speaker's name, whom your question is addressed to, and also your questions. So finally, after the Q&A session, for attendance and e-certificate purpose, please click on the links provided at the end of this session so that you can get registered for the attendance and e-certificate. And one more thing, we sincerely encourage participants to follow our social media profiles. This is because we will organize more webinars like this in the near future, and we like to share the good news with everyone before it happens. So you can follow both WUMBI as well as Codon Genomics website, Facebook account, and Twitter account as the following. So please, you can take part in our social network, and then yeah, you'll get informed about the latest development in the institute as well as Codon Genomics. So without further ado, let's start with our first speaker. So our first speaker is Dr. Nurul Shakima from WUMBI. Dr. Nurul Shakima is a molecular biologist working at UKM at WUMBI as a senior lecturer as well as the head of quality assurance. She is an expert in cancer genomics and has won a number of awards and grants. The topic for today is next generation sequencing of colorectal cancer in WUMBI, paving the road for precision medicine. Welcome, Dr. Shakima.
Assalamualaikum and good afternoon, everyone. First of all, I would like to thank the organizer for the kind invitation and to all of the participants for making time for me here today. I am Shakima from UMBI, and today I will be sharing what we have done with regards to next generation sequencing in colorectal cancer and how the works that we did could help to pave the routes for precision medicine. Firstly, for the basics of colorectal cancer. According to estimates from the International Agency for Research on Cancer in 2018, globally, CRC constituted approximately 1.8 million new cases and 9,100 deaths annually, making it the third most commonly diagnosed malignancy and the second leading cause of cancer death. Generally, industrialization and economic growth lead to a Western dietary pattern, sedentary lifestyle, and increase of obesity rates, all of which are major risk factors for CRC. Thus, the westernization of countries tend to be followed by a rise in CRC incident rates. This pattern is supported by higher CRC incident rates observed in economically developed countries. As cancer is a disease of aging, the rates of CRC development and death increase rapidly after age 50 years old, with an estimated 90% of global cases and deaths occurring after this age. The age-adjusted rates are higher in men than in women, yielding an approximately 1.4-fold and 1.5-fold difference for incident and death respectively, and the difference becomes more evident after the age of 50 years. The higher CRC rates in men might relate to a combination of multiple factors. Compared to women, men seem to be more strongly influenced by environmental factors than by genetic factors in colorectal carcinogenesis, with heritability of CRC estimated at 28% for men and 45% for women. In addition to their potentially higher vulnerability to environmental risk factors, men also have a higher exposure to such risk factors. For example, visceral fat, alcoholic beverage, smoking, and poor dietary patterns, but lower uptake of CRC screening based on fecal sample. In addition, men do not benefit from protective effects of endogenous estrogen as women do. Knowledge of these factors could help to inform prevention strategies in the future. The majority of CRCs, which is more than 90%, are adenocarcinoma subtype, which is a malignant neoplasm that develops from glandular epithelial cells of the colon and rectum. Approximately 60 to 65% of CRC cases arise sporadically, that is, it occurs in individuals without a family history of CRC or inherited genetic mutation that increase CRC risk, which is acquired through somatic, genetic, and epigenetic aberrations, largely attributable to potentially modifiable risk factors. CRC also has hereditary component, with twin studies having estimated the heritability of CRC to be around 35 to 40%. Approximately 25% of CRC cases have a family history of CRC without any obvious genetic cancer syndrome. Only 5% are attributed to hereditary cancer syndrome such as hereditary non-polyposis colorectal cancer, HNPCC, or also known as Lynch syndrome, or familial adenomatous polyposis, FAP, caused by inherited germline mutation in rare but high penetrance susceptibility genes, for example, the MLH1 gene, and APC gene respectively. Those genes identified from GWAS, the common but low penetrance genetic variation known to be associated with CRC risk, only explain less than 1% of CRC heritability. Please also note that CRCs with any heritable component are not fully hereditary because environmental factors could also contributing to carcinogenesis process. The mainstay of treatment for colorectal cancer is surgical resection, which offers the best curative outcome. The chemotherapy and radiotherapy are used to downstage as adjuvant therapy and also for palliative purpose. For the chemotherapy, the fluorouracil containing regimens are commonly used with or without combination with other drugs such as oxaliplatin, the irinotecan, or leucovorin. For a long time, doctors have been treating patients with the same disease using the same approach, the same drugs, and the same dose. The outcome of treatment have strongly suggested that the individual variation, especially the genetic variance which differs between individuals, must be taken into account, and the one-size-fits-all approach is no more valid for many diseases. Studies have also shown that anti-cancer drugs are effective in perhaps only 25% of cases, and that 6 to 8% of patients given medication will have adverse reaction. Therefore, the introduction of next-generation sequencing allows us to identify the right drugs or right treatment for precision medicine. Precision medicine is mainly about genome-driven clinical decision-making, which is the use of genome data from the patient to decide on tailoring the best treatment for the patient. The knowledge of the profile of this gene variant will allow the doctor to determine the type and the dose of medication that is suitable for each patient, because different patients need different drug or different dose. The technological advances in genome sequencing technologies and the cheaper cost has allowed more and more patients to be profiled at the molecular level. However, there are still a lot to be learned, but certainly we know more than before. In the case of cancer, the whole genome sequencing of tumor tissues has enabled us to understand that every tumor has its own molecular signature, which has both prognostic value, but also has allowed researchers to identify what is termed now as actionable or targetable mutations. Next, I will be sharing some of the NGS research on colorectal cancer that we perform in the UMB. Due to time limitation, I have only selected 3 studies in this topic. The first project that I would like to share is the LRGS project that I have been involved in since 2015. This program is currently headed by our Pro Vice Chancellor, which is Professor Rahman Jamal. This big program involves 5 projects from 3 universities. It covers the genome aspect of colorectal cancer, the proteomic aspect, the metabolomics angle, the integrated analysis component, and last but not least, the psychoeducation component among the colorectal cancer patients as well as breast cancer patients. We perform whole genome sequencing on 50 colorectal cancer patients and compared the variant with the normal adjacent colon tissue or the DNA derived from the patient's blood. On top of that, we also perform MSI testing with the hope to correlate the MSI status with the mutation burden and treatment outcome. From this study, we observed that each of our CRC patients have unique gene mutation signature. This signature has precision medicine utility. For example, KRAS mutation at exon 2, 3, or 4 are associated with resistance to anti-EGFR therapies. Loss of SMART4 is often associated with resistance to 5-FU chemotherapy, and inactivating mutations in FBXW7 interfere with the production of a functional FBXW7 protein, potentially conferring resistance to anti-EGFR monoclonal antibodies. The mechanism through which mutation in FBXW7 genes could impair cetuximab or panitumumab efficacy requires further investigation. It is possible that, since some of its proven targets are downstream effectors of EGFR, mutation of FBXW7 could impair their degradation and thus contribute to the resistant phenotype. The BRAF V600E mutation is often associated with resistance to anti-EGFR treatment in metastatic CRC patients with KRAS wild-type tumors, the sensitivity to BRAF inhibitor in melanomas but lack sensitivity in CRC, and sensitivity to combination therapy with BRAF and EGFR inhibitors. Now, for the next few slides, I will focus more on the microsatellite instability. The microsatellite instability, or MSI, is the expansion or reduction in the length of repetitive DNA sequence, known as microsatellites, in the tumor DNA compared to the normal DNA. Mutation in the genes for mismatch repair proteins is a key step in the adenoma carcinoma sequence, whereby when there is loss of 2 copies of MMR gene, such as MSH2, MSH6, PMS2, and MLH1, results in microsatellite instability. Why is MSI status important? Because MSI status of CRC provide insights into prognosis and treatment response. Evidence suggests that MSI high CRC, despite its tendency towards unfavorable histology, such as being poorly differentiated with mucinous histology, it has a better prognosis than non-MSI high CRC. So how does MSI relates to better prognosis? It was postulated that it was due to the presence of neoantigens, which are the peptides produced by tumors as a result of MSI-induced frameshift mutations. As novel peptides, neoantigens are highly immunogenic. This will lead to the infiltration of tumors with neoantigen-specific cytotoxic T cells and therefore inducing an anti-tumor immune response. Although MSI high CRC might not respond to adjuvant 5-fluorouracil-based chemotherapy, it is considered as a good candidate for immunotherapy targeting immune checkpoint modules such as PD-1 or CTLA-4 that suppress the T cell activity. We observed that almost all of the MSI-CRC were highly associated with high mutation rate, except for one sample. Among the 5 MSI-CRC patients, 3 of them display either stop gain, frameshift deletion, or non-synonymous mutation in mismatch repair genes MLH3, MSH2, and MSH6 respectively. We tried to relate the mutation profile, MSI status, and treatment outcome. However, this is where we face a little bit of challenges. The treatment outcome record in our hospital is not digitalized. Fortunately, we had the help from our collaborator in the oncology unit. Another thing is that not all of the patients underwent chemotherapy, thereby further limiting our sample size. In addition, only 10% of CRC patients in our study has MSI high. Furthermore, as you can see from the table, there is only one MSI high patient among the non-responders. The research is still ongoing and we look forward to a more promising and interesting result in the future. As a continuation from the previous ARGS project, we further explore the immunogenomics potential among our patients. This recently secured grant aimed to identify and validate the neoantigens via TCR-engineered T cells. We have just started this exciting project and we do not have results for now, but I'm going to share a little bit of background of this study. In CRC, tumors with MSI, as I mentioned previously, have more favorable response using immunotherapy treatments. The neoantigens, which is the new proteins generated by mutated cancer cells, may become better oncological indicators due to the longer response and greater specificity. Our previous whole genome sequencing data in CRC enabled us to predict the engaging neoantigens. These prioritized neoantigens will be then further validated in vitro culture of immune cells such as the monocytes and CD8 T cells. Next, the specific T cell receptor clone will be sequenced and subjected to transfection in T cells. The ability of the TCR-engineered T cells to recognize and kill cancer cells harboring the specific neoantigens will be determined. This study, as a whole, will further expand our in-house genomics data, as well as increasing our bioinformatics skills in detecting patient-specific neoantigens, especially among the Malaysian CRC patients. Moreover, this project will generate a proper and viable pipeline of converting molecular data to a translational approach. Furthermore, we will gain better understanding of tumor immunology mechanism, particularly in CRC. Another NGS-based colorectal cancer project that I'm involved in is a collaboration between UMBI with Newcastle University. For this project, I have had the opportunity to attend a 3-month attachment in the Newcastle, UK, and I was trained on the protocol by the first author of this publication. Basically, this assay, which is called single molecule molecular inversion probe and sequencing-based MSI assay, include 24 macrosatellite markers. These 24 macrosatellites were amplified in multiplex and trained using an in-house classifier based on 98 CRC samples. The sample classification achieved 100% concordance with the Promega assay in 3 independent cohorts with a total of more than 200 CRC samples. Furthermore, the assessment of assay detection limits shows that this 24 marker panel is able to detect as little as 3% high level of MSI DNA in sample mixtures and requiring a minimum of 10 templates molecule to be sequenced per marker for more than 95% accuracy. In addition, BRAF V600E mutation analysis was also included, with all of the V600E variants being correctly identified. These are all the people and funding bodies that I have to thank for all the support throughout the project, especially to Professor Dr. Iraman A. Jamal, which is the project leader for most of the colorectal cancer research project that I'm involved in, to the Ministry of Science and Technology for the grants, the Epigenome Grant and Science Fund, the Ministry of Higher Education for the High COE grants, ARGS as well as FRGS grant, the UKM for providing research facilities, all of our collaborators, especially from the Codon Genomics Sdn Berhad, from UITM, from UM, and also from MGI. My beloved team members, the UMBI Cancer Genome team, UBI Biobank for helping us a lot with the specimen collection. Not to forget our collaborator from Newcastle Universities, especially Dr. Jayanti, and all of my postgraduates who have been doing all the hard work to produce good results in this research. With that, I thank everyone for your attention, and I will be happy to take in any questions. Thank you.
Thank you, Dr. Shakima. So now let's move on to the next speaker. Our next speaker is Mr. Chua Subun from Codon Genomics. So Mr. Subun is an application scientist specializing in molecular biology and bioinformatics. He is currently a PhD candidate from University of Malaya,
你看到了？
working on several approaches in transcriptome analysis and various sample types. His topic is next generation sequencing and its application in medicine. Welcome, Subun.
Hi, thank you. Thank you for the introduction. So I'm Subun from Cordon Genomics, and I'm glad that to have a chance to be participating in this webinar together with Umbi. And today I will be sharing with you some knowledge regarding the next generation sequencing technology and its applications in medicine fields. So today I will be covering briefly about the different kind of next sequencing technology, and I will also bring up discuss some of the application that's brought by all this NGS technology. Yeah, if you look at the picture on the left, is a Encyclopedia Britannia. There's a total of 32 volumes of this encyclopedia, and they are actually more than 44 million words in this total of 32 volumes, and it contains 300 millions of characters. So the informations or topic covers include geography, biology, medicines, literature, physics, and astrology, and also religions. And on the right is actually the human karyotypes, and it contains 23 chromosome pair, and it has a total of 3 billion character per haploid genomes. And I'm sure that you all know that it contains every bit of information to make a human, from cellular to tissues and also organ level. So in order to understand the biology of humans and subsequently improve it, therefore we need to understand our source code of life, is the DNA or our genome. So therefore the technology to sequence it is very important. So the sequencing of the genomes can use to be answer questions like what is the code that are available in the particular organisms, how the code actually used to make the organisms, and what happened to the organism if the code gets changed through mutations. And the DNA code also is a cumulative record of our past, and through sequencing, it also helps us in understanding where do these organisms come from through their study of their evolutionary history. And we were also able to know how are these organisms related to each other. And the journey in the development of sequencing technology is always very fascinating for me. Along this way, a lot of Nobel Prize have been rewarded for the work related to it. It was started with the discovery of DNA structures by Francis Crick and James Watson, and afterwards, people are actually trying to develop sequencing technology that would be able to crack all these DNA code. And there are some of the point whereby breakthrough was brought to the sequencing technology. This the first one would be the when the Frederick Sanger actually developed a chain terminator inhibitor sequencing methods, and it was followed by the launch of a human genomes project since 1990 and the completions of the human genome project since 2001. And in 2003, there was actually a capillary sequencer that based on the Sanger technology that was launched by ABI, and this is actually include the throughput by a lot, but it's still limited. The throughput is still not compared, cannot be compared to the what is coming after that. This was through the launch of the first next generation sequencer 454 in 2005. And after that, there were a series of next generation sequencer that were launched after that. And currently, the Illumina's platform will be the most dominant. Actually, is dominated by market by now in the second generation sequencing or the next generation sequencing. And another recent breakthrough would be in 2011, whereby it was marked by the launch of third generation sequencing by PacBio Bioscience, which is actually also what we call a long read sequencing. And this is the zoom-in versions of the developments of the first generation sequencing. It started with the discovery of double helix system to the invention of Sanger methods, and eventually end with loss of the capillary sequencer, which is actually a highest throughput versions of the first generation sequencing. And the Sanger sequencings have a read length of around 800 base pair per reactions. And if you think that human genome is 3 billion base pair in size, so in order to sequence the human genome, we would require at least 3.75 millions of reaction. That is why the first human projects, first human genome project was actually took 10 years and $3 billion USD to sequence and assemble. So with the limitations in terms of the throughput in the first generation sequencing, there was always a need to the invasions of the next generation of sequencing with higher throughputs. So second generation sequencing was kicked off with the launch of 454 Pyro sequencer. And as I mentioned earlier, there were actually different sequencing platforms that were launched by different companies, but the current one would be mostly dominated by Illumina. And Illumina actually have a sequencer that from the sequencer from the benchtop type to the production scale sequencer that fit almost every applications and lab. And this is the general flow for a bacterial whole genome sequencing. So it started with a fragmentations of genomic DNA during a library prep, and the library prep also includes the editor ligation, which enable them to bind to a sequencing flow cell. And DNA library is then sequenced in the flow cell. So the short sequence will then be assembled back to the genome through the overlapping fragments. And all this could be completed within 3 days due to the high truths due to the availability of next generation sequencing. And if you look at this puzzle, which one would be actually easier to assemble? So of course it's a puzzle B side, because so the puzzle A usually resembles second generation sequencing, while puzzle B resembles third generation sequencing. When we have a short read, it creates challenges when we need to assemble it back, especially for large genome that is highly complex and with a lot of repetitive region. Therefore, there was actually a need for a long read sequencing. So as I mentioned just now, although the second generation sequencings come with a very high throughput, it also comes at a cost of the read length. Hence, this way the third generation sequencing come by, which offer actually a high read length with also very high throughputs. So it was started with the launch of PacBio I system in 2011, and followed by the improvement over the years in terms of accuracy and also throughputs. And here is a slide that gives you an overview of the various sequencing technology generation. So in very briefly, the first generation sequencing had the lower throughputs, and the second generation sequencing improved a lot in terms of the throughputs. While the third generation sequencing make improvement in terms of the read lengths, therefore allowing new applications such as isoform sequencing. So I hope I provided a brief view regarding 3 different generations of sequencing technology. And if you look at this slide, it's actually showing the field of study within omics. And inside the dotted line box is whatever we have inside an organism or cell. And the cell will respond to environments and produce observable phenotypes. The central dogma of molecular biology states that the DNA contains instruction to make a protein, which are copied by the RNA, and RNA then use the instruction to make a protein. The study of the DNA is genomics, study of RNA is transcriptomics, and study of protein is proteomics. And the study of gene regulations by DNA methylations and histone modification is epigenomics. And all these next generation sequencing actually can cater to all these study in the genomics, epigenomics, transcriptomics, and metagenomics. And NGS can be applied in different fields such as agriculture, forestry, food science, environmental science, social science, veterinary science, pharmaceutical science, and also, of course, medical science. And today I will be focusing on the applications of NGS on the field of medicines. And there are a few areas in precision medicine where NGS is playing its role. It can be applied in cancer research, identifies, identify and understand hereditary and rare diseases, and also to understand infectious diseases. It can be applied in research institutes such as IMRI or other every research lab. And once you have some outcome from the research lab, it can actually NGS can also be applied uh, in bringing the application to real life, whereby it can provide risk assessments, diagnostics, and also potential tailored treatments for patients. So when cancer was first described by the ancient Greek physicians Hippocrates, he identified only just two forms, the non-ulcer forming carcinomas and the ulcer forming carcinoma. So it's until the late 19th century, physicians found with the help of microscope that cancer have multiple cellular form. So now with the ability of next generation sequencing is again transforming our understandings of cancer origins and complexity. Instead of a broad categorizations based on the locations of tumor, genome sequencing is actually providing the detailed characterizations of the combinations of genetic mutation that triggers or aid cancer developments in an individual. So cancer affects more than 40,000 people in Malaysia in 2006, and number of cancer deaths was uh, enormous. And among the most prevalent cancers are breast, colorectal, and lung cancer. And it should be noted that the cancer can be caused by multiple gene mutations. For example, in the case of non-small cell lung carcinoma and SCLC, the cause could be due to more than 10 types of mutations. For instance, it can occur in EGFR genes, which control the protein called epidermal growth factor receptor, and this protein help your cell to grow and divide. And EGFR mutations is more common in women's people who don't smoke and those with adenocarcinoma types of and SCLC. It could also due to the mutation in uh, the it could also due to the mutations in the KRAS gene, whereby the genes play a role in making a protein called KRAS, and it help this gene actually help the cell to grow and also divide. So the question is how can NGS help in the treatments of cancer? It, it can play a role in from the beginning in terms of the help in the detections and also diagnosis. The tumor cell can be sequenced using NGS using cancer-specific panels such as breast cancer panel, colorectal panel, and also lung cancer panel. And follow from there, a somatic mutation specific to the patients can be compared to a database to identify the genetic aberrations. And with accurate detections and diagnosis, evidence-based action can be taken whereby the patient will be recommended to undergo targeted therapies or immunotherapy. In case whereby there is no available approved drugs for the genetic mutations detected in the patient, it is recommended to be facilitated to registration for the clinical trials. So the sequencing technology also evolved over time, and recently the sequencing of full isoform is possible with the availabilities of isoform sequencing or isoseq with PacBio platform. So in this study, it was used to understand the therapeutics resistance in CRPC or castration-resistant prostate cancer, and targeted smart sequencing of this androgen receptor AR isoform revealed that the structure of ARV9 was previously mischaracterized and have omitted a cryptic exon that was thought to be appears only in ARV7. So ARV7 has been identified as a potential biomarker for drug resistance based on knockout experience targeting both isoform. So this data actually suggests that ARV9 may in fact be a more predictive isoform, and this finding could be attributed to the availabilities of isoseq from PacBio. This is show that we have actually have all kind of sequencing technology that could actually fit every application, and all these sequencing technology will evolve over time to actually help us in our biological research. Next, I will talk about how NGS help in identifies and understand rare diseases. The images actually show a chorea dance which is actually related to people having Huntington disease, and they will suffer contortions of their bodies, and during the Middle Ages, many innocent victims were persecuted and even tried at witches. And this show the danger of not having understandings of disease. And this is a case study about a girl named Hannah Mannings, and she has actually one of the rarest disease in the world. So at the age of 2, she had her first seizure, and at 4, she was diagnosed with autism. When she was referred to doctor, genetic test came back as normal, and she also later shows so some of the syndromes like cognitive deficits, muscle weakness, exercise intolerance, etc. So this until eventually only through a whole exon sequencing revealed that she actually have a gene mutation that caused the spinal cerebellar ataxia, autosomal recessive 9 or SCA9. This shows the importance of having NGS as a genetic tool, as it offers a more sensitive and precise disease detection that allows us to detect even the rarest disease. This next example shows one of the success keys of using NGS as a diagnostic tools. Cystic fibrosis is a progressive genetic disease that causes persistent lung infections, and it affects also the digestive system. This is most common among the Northern Europeans, and it affects 1 in 3,000 newborns, and 1 in 25 is actually a carrier. There's no known cure, but the newborns earlier diagnosis and the treatments can actually improve their growth, keep their lung healthy, and also reduce the hospital stay and eventually add years to their life. Carrier testing also allows parents to find out what is the chance of having a child with CF, and this will eventually affect their family planning decisions. So this CF disease is actually caused by a recessive gene, whereby if both parents are carrier, there's a 25 chances that the child will born with CF. Because of the severity of the disease, detections kit have been developed for the detection of this disease. The kit available from Illumina can actually be used with a MySeq-Dx sequencer to identify the genetic mutation, and this kit is actually FDA cleared and also CE ICD market. And This show a real case on actually how NGS can be applied even at the stage of diagnostics and not only in a common research lab. Yeah, in our past history, infectious disease was always feared due to the lack of information and superstitions. And miasma theory was the predominant theory of disease transmission, whereby the disease such as cholera, chlamydia, or Black Death were caused by miasma, or is also termed as a nauseous form of bad air. It was widely held until the germ theory was popularized in the late 19th century, and since then, microscope was used to understand pathogen. In recent decades, NGS offer an even more precise method to understand pathogen, whereby it can be used to sequence the genome. In 2020, a patient was confirmed to have a pneumonia through the CT scan, and Burko-alveolar laboratory was extracted from the patient's lung, and after that, the RNA was extracted, and a deep metal transcriptome sequencing was performed. It was then followed by de novo assembly to acquire the virus genome. Yes, this is actually the first whole genome sequencing of SARS-CoV-2 virus, and I'm sure everyone will know about this. Yeah, I'm still very impressed by the speed where they're able to isolate, sequence, and publish this paper in less than a few weeks. And this speed would be unimaginable just 20 years ago. And followed by this whole genome sequencing test kit was actually designed and actually used for the mass testing. Yeah, this is another case study whereby NGS could identify the association between microbiota and also gastric cancer. And this employ a 16S metagenomic sequencing to compare the gastric mucosa biopsy sample between cancer patients and healthy control. And out of this, they could actually identify a few microbes that are actually risk factor, such as Pylori, P. acnes, and P. copri, and they also are able to identify the protective microbe, such as Lactococcus like this. So here I have a very simple summary regarding how actually NGS can affect research and also bring impacts to the medicines. So as I said earlier, NGS can be applied in research lab, and also the outcome from the research lab can be actually used to bring direct impacts in our health system, such as provide a risk assessment, diagnosis, and also potential tailored treatments. For example, in field of oncology, it could be applied to identify new cancer markers, identify the new type of somatic mutations, and based on the result, risk assessment can be performed on other persons, so to understand such as the risk of cancer based on their susceptibility, and also in terms of diagnosis, it could be identified what kind of somatic mutation that a patient has, and tailored treatments based on the mutations could be performed. And in terms of hereditary and rare diseases, of course, research, the NGS could be help us to understand the disease in the molecular levels, and it could help us in have a risk assessment during, let's say, pre-marital genetic counseling. In terms of diagnosis, before we do IVF, we can have a pre-implantation diagnosis using NGS, and the treatment could be based on more specifics based on the specific type of your disease. In terms of pathogens and infectious diseases, NGS can be applied to identify causative pathogens, identify host pathogens relationship, and it's actually also can be helped to provide risk assessment for patients, as we can monitor their gut health and risk of developing diseases using NGS. And of course, this I mentioned in one of my earlier case studies in the sequencing of SARS-CoV-2 virus. It can be used to diagnose causative pathogens, and it could also act as to assist the conventional diagnostic methods. And the end of this will be it help us to provide a more precise diagnosis, and eventually it could lead to a more precise. Let's say if you found, if you can identify the precise identity of microbe, you can actually prescribe some more antibiotics, so that you won't have problems such as antibiotic resistance. So we at Gordon Geomi, we also offers all these kind of sequencing, next generation sequencing technology that would actually helps from in helping researcher to do their work in their labs, and at the end they can actually apply all this to actually bring impact to the healthcare system. Yeah, so that's the end of my presentation. So let's work together to create a better future using data, science, and social technology. Thank you. Here, I think I will pass back to the moderator.
Thank you, Subun. So yeah, I would like to remind the participants that actually, if you have any question, you can actually start asking questions via commenting through Facebook or through the Q&A feature in the Zoom platform. So our next speaker is Dr. Aida Abdullah from UMBI. Dr. Aida specializes in epidemiology and biostatistics and is heavily involved in the Malaysia cohort. She obtained her PhD from Newcastle University, Australia in 2018, and her topic for today is type 2 diabetes, a predictive model using GWAS, a diverse Malaysia cohort population study. Welcome, Dr. Aida.
Thank you, Dr. Lau, for your kind introduction. Assalamualaikum and a very good afternoon to all audience. Today, I'm going to share our experience on analyzing GWAS diabetes using the Malaysian cohort data. Type 2 diabetes is a complex polygenic disease that become pandemic globally and locally. Malaysia has the highest comparative prevalence among Asian countries. The prevalence of type 2 diabetes in Malaysia almost doubled up within a decade. In addition, National Health Mobility Survey 2019 indicate that 1 in 5 Malaysian adults were living with diabetes. There were also disparity in prevalence of type 2 diabetes among our Malaysian cohort participants, which highest in Indian, followed by Malay and lowest in Chinese. The Malaysian cohort is a national project that initiated by Prime Minister and successfully recruited 106,527 Malaysian adults aged 35 to 70 years from April 2006 until September 2012, all over Malaysia. Currently, the Malaysian cohort is in follow-up phase since 2013. The Malaysian cohort also serves as a platform for big data analysis and largest resource of biobank sample in Malaysia. For those who are interested more on the Malaysian cohort, you can refer to our publication and website. There have been 3 main approaches employed to identify genetic risk variant for common complex disorder. They are linkage study, candidate gene association study, and genome-wide association study. Familiar linkage study identify disease and genetic marker segregation through multiple generation of family. It is challenging to do familiar linkage due to difficulty of identifying multiple generation of families with disorder of interest, limited mapping power, inability to precisely localize underlying risk variant, and also costly. In contrast to linkage study, the candidate gene approach search for association between common genetic variant and disease, restricting to pre-specified genes of interest. Candidate genes are typically selected based on a prior knowledge or hypothesis reflecting the gene's presumed biological role in disease. The candidate genes approach has been limited to small sample size, restriction of hypothesis to non-biology, and inability to replicate many results. At the time of conducting this study in 2012, GWAS has been emerged as the method of choice of identifying common genetic variant in complex diseases. GWAS was facilitated by completion of Human Genome Project in 2003, the International HeadMap project in 2005 that catalogued millions of common SNP and the development of high-throughput genotyping array. The first notable GWAS was conducted in 2005, identifying age-related macular degeneration. Earlier linkage study found 4 genetic loci linked with type 2 diabetes. However, only HNF4A has been confirmed by GWAS. While numerous variants has been reported associated with type 2 diabetes using candidate gene approach, but only 3 loci were robustly confirmed by GWAS. The first type 2 diabetes GWAS was published in 2007, and as far for January 2020, more than 100 publications on type 2 diabetes and its complications listed in online GWAS catalog. GWAS can identify genotype-phenotype association of complex disease. It requires no prior knowledge about the causal gene, such as the gene function and their location on the genome. It can also dissect the biological basis of underlying disease as well as study low frequency and rare variant. It also can be used for personalized medicine and development of new drugs. The initial type 2 diabetes GWAS were all restricted to population of European ancestry. The first 2 large-scale type 2 diabetes GWAS conducted in Asian population were reported in 2008. This study demonstrated the utility of extending GWAS to non-European population where KCNQ1 variant associated with type 2 diabetes in Asian population was not previously detected among European ancestry. A subsequent 2011 meta-analysis included Malay, Chinese, and Indian population from Singapore. This study alone contributed an additional 16 novel loci in spite of relatively modest sample size. This is a nested case control study that involved 4,077 participants from the Malaysian cohort study, which consists 1,323 Malay, 1,344 Chinese, and 1,410 Indian. Cases were defined as those with fasting blood glucose more than 7.5 millimole per liter, while control less than 5.5 millimole per liter. Our aim of the study is to investigate the association and relative contribution of genetic variant to the risk of type 2 diabetes in the 3 Malaysian population group, which is Malay, Chinese, and Indian. We also wanted to assess the influence of gene-environment interaction on the risk of type 2 diabetes. GWAS workflow involves few steps before we can analyze the result. In our study, we genotype our sample using metabochip array from Illumina that consists of 196,725 variants from cardiometabolic disease trait. Then, we did quality control or data cleaning at sample and SNP levels before we proceed to statistical analysis that involve test employed. Quality control assessment at sample level include concordance rate, call rate, sample-dependent control, and also gender validation. Concordant rate assess the accuracy of genotyping. The score of 1 indicate perfect genotype, while score less than 0.3 indicate non-duplicate sample. Next, we assess the genotyping call rate. This call rate was generated by Genome Studio software, and we filter out the samples that has call rate less than 0.95, like shown in this plot. Then we look at sample-dependent control that indicate the specificity and the sample quality. Good quality sample will give the result like figure A, where all the signal at the background. Stringency control assess the stringency of hybridization process and identify the presence of non-human DNA. This is because the probe are complementary to bacteria sequence. Figure A show a good quality sample. There are several filtration processes for quality control at SNP level, such as minor allele frequency, missingness, Hardy-Weinberg equilibrium, heterozygosity, cryptic relatedness, and also cluster plot. SNP level QC was done using peeling, a whole genome association analysis toolset that freely available in the internet. We filter out minor allele frequency that less than 0.01. Some study remove minor allele frequency less than 0.05, which is depend on the available sample size. Then, we filter out SNP missingness that more than 0.05 and divergent from Hardy-Weinberg equilibrium at p-value of less than 10 to the power of -6 in control. We also remove outlying heterozygosity that has ±8 standard deviation from the mean and cryptic relatedness more than 0.1875. We also filter out SNPs that are not well clustered, like in the figure A. SNP with high quality should show distinct, well-separated genotype, like in figure B. As for analysis, we did logistic regression, QQ, and Manhattan plot to assess the association between SNP and type 2 diabetes. We also performed principal component analysis and genomic control to assess population stratification effect. We also did multiplicative and additive interaction to assess interaction between gene and genetic risk score and lifestyle risk factor. The QQ plot here show the p-values for association of 62 SNPs observed in each ethnic group and meta-analysis with type 2 diabetes. This plot clearly show the p-value were more significant than expected value under the null hypothesis. This Manhattan plot from meta-analysis of 3 combined ethnic groups in Malaysia indicate the significant p-value after multiple Bonferroni correction. The x-axis represents the genomic order of chromosome, while the y-axis represents the minus log time of the p-value of each marker. We use principal component analysis or PCA to study geographic variation in allele frequency using HeadMap and Singaporean Genome Variation Project. It is interesting to know that Malaysian Malay and Chinese are similar to Han Chinese from Beijing in HeadMap, while Malaysian Indian was similar to Gujarati Indian from Houston, Texas in HeadMap. Our population are almost similar with Singaporean population, as you can see in the right plot. We found 7 SNP that reached GWAS significant level, which is TCF7L2, CDKN2A, FTO, PPARG, GCK, MC4R, and ADCY5. And 10 additional variants that reach nominal significance at P less than 0.05. This variant involved in glucose utilization, pancreatic beta cell development and function, fatty acid concentration, insulin availability, and also obesity. Interestingly, PPARG that found in our population is targeted for anti-diabetic drug, tiazolidinone, with suggesting for personalized medicine in Malaysia. However, the genetic risk score that consists of 62 SNPs only contributed to less than 2% of type 2 diabetes with small effect size, ranging from 1 to 1.2. This result also suggesting that the soaring prevalence of type 2 diabetes in Malaysia is unlikely solely result from common genetic variant. Our lifestyle predictive model risk factor indicate 6 significant risk factors including physical activity, waist circumference, age, body mass index, gender, and waist to hip ratio. All these factors contributed to 21% of type 2 diabetes in Malaysia. The combination of genetic risk score and lifestyle risk factor model only explain additional 1 to 2% of type 2 diabetes risk factor across the population. There were also 10.6% of the total 310 interactions that reached nominal significance. It seems that the strongest interaction were between BMI and 3 variant, which is CDKN2, IGF2BP2, and LPP. This first plot shows an evidence of genetic risk score gradient effect across strata of BMI, with the GRS having progressively larger effect with decreasing level of BMI. This finding is similar with EPI-Interact case control study. In addition, this result also consistent with our interaction result in the previous slide. In conclusion, we found the relative effect of common genetic risk variant appears small. However, there was genetic risk score gradient effect across BMI strata observed. We also found 33 individual SNP lifestyle interaction that reached nominal significant p-value, suggesting that true gene lifestyle interaction for BMI may exist. Recent changes in lifestyle and diet contributing to abdominal obesity with aging population are major contributor to the increased prevalence of type 2 diabetes in Malaysia. Targeted public health intervention focused on reducing obesity will help to mitigate the soaring prevalence of type 2 diabetes in Malaysia. The potential of GWAS is still underexploited due to methodological limitation. Many challenges have emerged, including detecting epistasis, SNP with small effect, and missing heritability. The issue have motivated advancement in GWAS analysis in both statistical modeling and machine learning. These are the recent software in GWAS with their targets, inputs, and analysis method. Further studies that present a direct, greater comparison between different methods in terms of performance and reproducibility of GWAS analysis will help to enhance and fully exploit the potential of GWAS. These are some publications that come out from TMC Diabetes GWAS study. With this,
Thank you, Dr. Aida. So next, we have Ms. Lau Cheng Yi from Codon Genomics. So Ms. Cheng Yi is a data scientist from Codon Genomics. She's very experienced in data mining, statistics, and bioinformatics. And her current research focus is on the modern data science, specializing in predictive explanatory model building. Her topic is machine learning in medical diagnosis, a case study with Wisconsin breast cancer data set. Welcome, Ms. Cheng Yi.
Hi everyone, I'm Cheng Yi, a data scientist from Codon Genomics. Today, I'm going to demonstrate the use of machine learning technique by developing a predictive model for breast cancer diagnosis using a breast cancer dataset collected by University of Wisconsin. The data is available online at UCI Machine Learning Repository. It consists of 569 samples with 32 attributes. The features are computed from a digitized image of a fine-needle aspirate of breast mass. They describe characteristics of the cell nuclei present in the image. Here is the attribute information. The response variable is diagnosis, with 312 matrilineal and 357 benign class labels. The mean, standard error, and worst mean of the three largest values of the following features were computed for each image, resulting in 30 features. The 10 real value features are computed for each cell nucleus: radius, texture, perimeter, area, smoothness, compactness, concavity, concave point, symmetry, and fractal dimension. Here are the first three rows of the Wisconsin breast cancer dataset. This is the ID number. This is the response variable diagnosis, and the others are explanatory variables with three groups: mean, standard error, worst, which compute from 10 real value features. For example, one of the real value feature, radius, we have mean of the radius, standard error of the radius, and worst of the radius. The objective of this study is to observe which features are most helpful in predicting malignant or benign cancer and to see general trends in the Wisconsin data. We aim to build a model that can predict whether a feature breast cancer sample is benign or malignant using a machine learning classification method. The initial step in this study is data exploration and visualizations. Data exploration, also known as exploratory data analysis, provide a set of simple tools to achieve a basic understanding of the data. The result of data exploration can be extremely useful in grasping the structures of the data, the distribution of the values, the presence of extreme values, and interrelationship within the dataset. The bar plot here shows proportion of the benign class, 62.7%, is higher than malignant class, 37.3%. Here are the histogram of mean of the 10 real values feature for benign and malignant class. Most of the features have higher mean and larger variance of mean in malignant class. Radius mean, perimeter mean, area mean, concavity mean, and concave point mean seems to be promising predictors since the overlapping region of histogram is smaller. Here are the histogram of standard error of the 10 real values feature for benign and malignant class. The overlapping regions of the histogram are big, and therefore these features are not so helpful for predictions. Here are the histogram of worst of the 10 real value features for benign and malignant class. They have similar pattern of distribution with those mean values group of feature. Similarly, radius worst, perimeter worst, area worst, concavity worst, and concave point worst seems to be promising predictors since the overlapping regions of histogram is smaller. Principal components analysis was performed to explore the high-dimensional Wisconsin breast cancer dataset. Clustering of samples by diagnosis class was observed in this space of principal component 1 and 2, which explain around 44% and 19% of the total variation respectively. Principal component 1 primarily separate the diagnosis class benign from malignant. Hence, the feature with strong loading on principal component 1 are potential predictor of diagnosis class label. The directed arrow here in the plot are loading vectors for top 5 features with the highest absolute loading values for principal component 1. This is the Pearson correlation metric plot which shows the increasing or decreasing linear trends of each pairwise feature in the data. The area radius parameter feature are highly positive correlated, which makes sense since area and perimeter are functions of radius. Thus, these information are considered redundant, and one of them is sufficient for indicating the size of the cell nuclei in the predictive model. This is the list of 2 sample student t-test statistics. Technically speaking, the t-statistic is the normalized difference of group average. Hence, the features with high absolute value of t-statistic are potentially useful predictors. The 2 features, perimeter worst and concave point worst, are selected for breast cancer diagnostic predictors. These features have the highest absolute t-statistic, strong loading on principal component 1, and small overlapping region in histogram. This process is called feature selections. The goal is to select a subset of the feature from the original dataset that reduce its dimensions by using a minimal set of features to represent the maximum amount of variance in the data. This subset of features is used to train the model. Feature selection serves 2 main purposes. First, feature selection often increases classification accuracy by eliminating irrelevant, redundant, or highly correlated features. Second, it decreases the number of features, which makes the model training process more efficient. Note that we can select multiple different subsets of reasonable features for developing multiple models. Then we can compare the performance of these models and select the optimal model for operations. Here we only demonstrate building a single model. There are many machine learning methods available to predict the class of a new data point. For example, support vector machine, naive Bayes, decision tree, random forest, and logistic regressions. Here we can build several models using different machine learning algorithms and compare their performance. Here we demonstrate how to build a single model using logistic regressions, which has the advantage of being relatively simple to perform and interpret. Categorical variables are commonly used in biomedical data to encode a set of discrete states, such as whether a drug was administered or whether a patient has survived. When the dependent variable is categorical, a common approach is to use logistic regression. A method that takes its name from the type of curve is used to fit data. Logistic regression is a powerful tool for predicting the class of the variables, e.g. a patient is survived, and the associated class probability, e.g. the patient has a 75% chance of survival. Logistic regression is a specific type of generalized linear model. It models the log-odd ratio as a linear combination of the predictor variables, as shown in equation 1. We arrange equation 1 and obtain equation 2. We classify a data vector x as belonging to some class y when probability for y given x is greater than some threshold value c. For this study, the data vector x corresponds to perimeter worst and concave point worst, and y corresponds to the response variable diagnosis. Thus, we get these equations. The class of breast cancer is predicted as Mendelian, when the probability of diagnosis is McLeanian greater than some threshold value C. Logistic regression is found iteratively via maximization of the likelihood of the observed data. In other words, the parameters of the logistic model are fit using the method of maximum likelihood. Logistic regression was done using the open source R statistical programming environment. Here is the output of the logistic regression analysis. The estimated coefficient correspond to the beta parameters of the logistic model. Here are the standard error for the estimated coefficients, and here are the z-value. All the p-value are well below 0.001, and thus all the estimates are statistically significant. We plug in these estimate coefficients and get these equations. The trained logistic model. Now we have done a simple logistic model. So, how well this model fit the data? Deviance is often used as a measure of goodness of fit in statistical modeling. The smaller the residual deviance, the better the fit. Deviance residue are analogous to the residue in linear regressions and can be used to check the model fit at each observations. They can be used to identify outliers. Deviance residues that are relatively far from zero may be outliers. The deviance value here can be used to calculate the McFadden pseudo R-square, which is analogous to R-square in linear regressions. Calculating the McFadden pseudo R-square will result in a value between 0 when the fit is bad and 1 when the fit is good. The McFadden pseudo R-square for this model is 0.8, indicating a good fit. Even the model has a high goodness of fit statistic, but we do not know whether this fit is overfit. The best form of assessing the propriety of a fitted model is generalizabilities, the ability of a fitted model to accurately describe or predict new data. An overfitted model has overly memorized the data set it has seen and is poorly generalized the learning to an unseen data set. That's why an overfit model results in poor test accuracy. Archaic information criterion is a fine technique based on in-sample fit to estimate the likelihood of a model to predict the future value. In plain words, the AIC is used to estimate the relative performance of a model when predicting new data. It estimates model relatively, meaning that AIC scores are only useful in comparison with other AIC scores for the same dataset. A lower AIC score is better. Since we only have a single AIC score here, it is not useful here to to have some information. Since we only have a single, as a reminder, AIC only measure the relative quality of models. This means that all models tests could still fit poorly. As a result, other measures are necessary to show that the model results are of an acceptable absolute standard. Cross-validation is a technique to assess the generalizability of a model to unseen data. The basic form of cross-validation is k-fold cross-validations. The dataset is randomly split into k-stratified folds. Each fold is used as a test set once, while the other folds are temporarily combined to form a training set for model generations. Performance metrics on the test set are calculated and stored, and the process is repeated for the number of folds that have been generated. To obtain reliable performance estimation or comparison, a large number of estimates is always preferred. In K-fold cross-validation, only K estimates are obtained. A commonly used method to increase the number of estimates is to run K-fold cross-validation multiple times. The data is reshuffled and re-stratified before each round. This technique is called repeated K-fold cross-validation. For this study, three-fold cross-validation was performed 8 times to obtain reliable performance estimation of the trained logistic model. The trained model was able to classify the breast cancer with mean accuracy 0.94, mean sensitivity of 0.95, and mean specificity of 0.92. Thus, the trained model has done a pretty good job. An ROC curve, receiver operating characteristic curve, is a graph showing the performance of a classification model at all classification thresholds. This curve plots two parameters, true positive rate and false positive rate. AUC, area under the curve, is an aggregate measure of performance across all possible classification thresholds. In other words, it measures the quality of the model's prediction irrespective of what classification threshold is chosen. The AUC here is 0.986, which is very high, indicating that the trained logistic model has very good predictive power. For your information, AUC is often used to compare the performance of multiple models using different machine learning algorithms. As a reminder, the breast tumor can be classified as malignant when the predicted probability is greater than some threshold value C. By default, C is equal to 0.5. This is the default threshold 0.5 with sensitivity 0.92 and specificity 0.95. The threshold 0.3 with the highest sum of sensitivity and specificity is also plotted. This is the best threshold defined in R. The threshold 0.3 has higher sensitivity 0.97 and lower specificity 0.92 than the default threshold. In our context of study, we might prefer lower false negative rate. That is, we try to avoid predicting patients with malignant tumor as benign. In this case, threshold 0.3 with higher sensitivity can be chosen for classification. Lastly, here is a graph that shows the predicted probability that each tumor is malignant along with their actual diagnosis status. Most of the malignant tumor, that one is in red, are predicted to have a high probability of the tumor is malignant. And most of the benign tumor, the one in blue, are predicted to have a low probability of tumor is malignant. The gray dashed line are the threshold at 0.5 and 0.3. If we set the threshold at 0.5, the sample above this line are classified as malignant. If we set the threshold at 0.3, the sample above this line are classified as malignant. For threshold at 0.3, there are more samples classified as malignant. This reduces false negative rate at the expense of slightly higher false positive rate. In summary, this study has identified two features, perimeter words and concave point words, for breast cancer diagnostic prediction. A logistic model has been developed and cross-validated. The trained model was able to classify breast cancer with high accuracy 0.94, sensitivity 0.95, specificity 0.92, and AUC 0.99. The optimal classification threshold with highest sum of sensitivity and specificity is 0.3. It has higher sensitivity and lower specificity compared to the default threshold 0.5. Let's conclude my presentation. Thank you.
Thank you, Ms. Jingyi. So now we come to the next speaker. So the next speaker is Dr. Muhammad Irfan from UMBI. So Dr. Irfan joined UMBI in late 2020 as a medically qualified statistician. Prior to joining UMBI, Irfan finished his PhD in mathematics at Newcastle University, UK, and at the same time he was trained as a medical doctor in Queen's University of Belfast. He is also a fellow of the Royal Statistical Society UK. His main research interest is in Bayesian statistics, particularly with respect to prior elicitation and Bayesian approaches in handling missing data. His topic is the Bayesian paradigm in modern data analytics. Welcome, Dr. Irfan.
The Bayesian paradigm in modern day data analytics. This is the outline of my talk today. Firstly, I will explain about the motivation behind this talk, and then the current statistical question and approaches that we face on day-to-day practice in scientific research, and criticisms on frequentist methodology and what we mean with a Bayesian paradigm and then recent Bayesian methodological developments. Up till now, we think that the current statistical paradigms can be divided into two, which is the frequentist school and the Bayesian school. We usually use a frequentist paradigm in terms of our data analysis, which is involved with the computations of p-values and the 95% confidence intervals. But our ideas about probability and statistics may be deeply entrenched, perhaps because of the widespread use of p-values and the confidence intervals in presenting our results to the medical journals. And the only and that this talk is aimed to broaden our horizons a bit. In most of our scientific endeavors, scientific research endeavors, we are mainly concerned about four types of statistical questions. The first one is your point estimation problems. For example, to gauge or to estimate the prevalence of diabetes in Malaysia. And then the other one is to measure the uncertainty of our point estimates, which is involved with the interval estimations. And then we want to predict based on our models that we develop, which is the prediction problems. And then we want to test our hypothesis, the hypothesis testing problems. So both frequentist and Bayesian school, we try to address the same problems. However, they use different methodologies. For example, for frequentist, for frequentist school, they use least square methods in linear regression. For example, for point estimate to address the point estimation problems. And then for logistics, for example, they use the MLE, the maximum likelihood estimation for point estimation as well. And then the confidence interval for to measure the uncertainty over the estimates, and the test statistics and p-values to for hypothesis testings. Bayesians, I want to emphasize this, Bayesians don't use all these family methods. The brief overview about the Bayesian statistics is because of the work of Reverend Thomas Bayes, was published in 1764 by his friends Richard Price 3 years after his death. The publication was entitled. The title of that publication was "Solving a Problem in the Doctrine of Chances," and this was published after his death by his friend Richard Price in Philosophical Transaction of the Royal Society of London. And because of this work, the Bayesian theorem became available to most people who are interested to use as subjective probability in their research method. However, there were not many proponents of these statistical methodologies, because most people, they were entrenched in the classical probability at the time. Frequentist school was not fully developed before World War II. But during Reverend Thomas Bayes, but during World War II, there was a cryptanalyst, Alan M. Turing, a mathematician who works at Bletchley Park, who successfully decrypted the Enigma code made by the Germans to transfer their coded message to their military personnel all over the Europe at the time. And Alan M. Turing used the Bayesian techniques to test computing hypothesis at that time, and because of that, he successfully managed to break the Enigma codes. This is the picture of Alan M. Turing that I obtained from the Wikipedia. So I would like to criticize the frequentist approach that we use at the moment, mostly in the literature. But and to do this, I have to use an example to illustrate such criticism. Suppose we want to run an experiment to see whether there's a difference between the BMIs, the population BMIs between the Fakulti Perubatan and Fakulti Sains Kesehatan student and staff. And then when we use a two-sample t-test, and we often test the null hypothesis, which is there's no difference between the two population groups, which is H0. The population BMI for the Fakulti Perubatan is equivalent to make a population BMI, the mean population BMI of the Fakulti Sains Kesehatan. So how can we test such hypothesis? So I generated a data set, a random data set using R, and then there were like 12 observation in the Fakulti Perubatan, and then another like 9 observation for Fakulti Sains Kesehatan. So the ratio is not equal for both groups, just for illustrative purposes. And from this, we can obtain the descriptive summaries for each groups. And then we can see that the sample mean for Fakulti Perubatan is 28.17, and then the sample mean for the Fakulti Sains Kesehatan group is 28.17. And because we assume that the standard deviation can be pooled, so we use the following formula, and then the pool standard deviation is 4.099. Okay. So we use the test statistics, and then we obtain the t-value as 2.147, and using R with a degree of freedom of 19, we refer to the t distribution table. We use R function there, and then we obtain the p-value 0.00448, or you can use a t distribution table. Therefore, at 5% level, we reject the null hypothesis, and then we can conclude that there is a significant difference between the mean BMI of these two populations. But the catch is, if you work at the 1% level, if you reduce your significant levels to 0.001, then you have to accept, you will fail to reject the null hypothesis. I don't use the word accept. You fail to reject the null hypothesis, because the null hypothesis can be anything. I mean, it can be some values. So when we don't reach the significance threshold, we can only say that we failed to reject the null hypothesis. So this indicates the arbitrariness of fixing the level of significance at 0.05. So another question is, what is the real interpretation of p-values then? And even the biggest proponent of p-values less than 0.05, who is the great Ronald Fisher himself, the R.E. Fisher, the founder of the Fisher exact test, he said in his books, in the Statistical Methods and Scientific Inferences, published in 1956, that however, the calculation is absolutely academic. For scientific worker, there's no such thing as a fixed level of significance, and the level of significance should be fixed according to hypothesis and according to the individual studies and according to individual circumstance. Because if we always fix the threshold values at 0.05, then there'll be in some particular cases that we might falsely reject the null hypothesis. I will come to that later. So these are the real interpretation of p-value. P-value is just like a measure to see the compatibility of your data with a specified hypothesis. In this case, you assume that your null hypothesis is true if only chance is the cause of such discrepancy. And based on Westerstein and Lazar 2016, I mean, based on the statements by the American Statistical Association, ASA, there are several grave p-values misconceptions in terms of the interpretation. It actually doesn't measure the probability of your hypothesis, or in this case, your null hypothesis being true. And then it doesn't measure the probability of your data being produced due to chance, random chance alone. It's actually, it doesn't give you the probability of the mechanisms that generate your data. Of course, the mechanism is because of the chance that generate your data, but it doesn't give you the probability how much influence that the chance generates your data if you think your null hypothesis is true. And the most important thing is we should reject our null hypothesis when p-value is 0.05, which is wrong, because there are other factors that we should consider. For example, the validity of assumptions that we use when analyzing the data. For instance, if we, in linear regression, there's an assumption there that we have to meet. And then one of the assumptions is there's heterodicity of the residuals, or the equivariance of the residuals. If the residuals, if the assumption is not met, then the p-values that we obtain from the analysis will be much smaller, or at least smaller than the actual p-values. And because of that, we may falsely reject the null hypothesis because the heterodicity assumption is not met. Okay, this is the interpretations of a confidence interval. And another, for example, there's another objective for this illustration. And then we can also compute the 95% confidence interval for the population mean BMI for faculty perubatan staff and students. So we use this following formula. And then we use the T statistics instead of Z statistics because the sample size is small. So we assume that distributions as a flatter tail. And then because of that, we cannot use the Z distribution or the normal distributions for our inferential purposes. And because of this, we obtain, after we do the computation, we obtain the lower limits of our 95% confidence level as at 25.28, and then the upper bound as 31.05. So does it mean that the probability that the true population parameters, or in this case, the true population mean of the BMI for faculty perubatan personnel and student is between 25.28 and 31.05? And is the probability is 0.95 or 95%? No, the answer is no. In the frequentist approach, population parameters in this case are not random variables. This means the population parameters or the population mean is fixed, but it's unknown quantity. So with your single confidence interval, the probability is either 1 or 0. But using a Bayesian framework, we can obtain an interval that truly reflects the 95% probability of capturing the population parameters. Okay, this is an illustration or example that I obtained from tan and tan 2010. For example, in your single interval, the confidence interval here, the single confidence interval that you construct, it can be either one of the 19 confidence intervals that capture the true population mean, or it also can be one of the 20, the ones in the red box, the confidence intervals that doesn't capture the true population mean. So when you construct confidence interval, it doesn't really reflect the uncertainties about your population parameters. It doesn't give you the probability of it. So in Bayesian framework, we shall see that Bayes' theorem gives us a way of combining our subjective assessment with observed data. For example, before we conduct the experiments, I would say the experiments before, we can have like a subjective idea about what we believe about the population parameter or of the BMI in one of the groups. What if from a previous study, somebody already published that in the literature, that we know that the faculty probatan group members were quite likely to have a BMI somewhere between 25 and 33, which means that we have a prior knowledge about it, about the true population parameter, how we incorporate that into our analysis. Will it then be insensible to use such information and combine with our data, and then we can update our prior information into something much more informative? And then a Bayesian analysis could do this. And this is in line with the inductive ways of doing scientific experiments. Usually, an experimenter usually knows something about the parameters that he or she wants to estimate, and then he or she carries out the experiment, in which the data will be collected, and then he or she updates his or her belief from the results. And then in Bayesian way, we can say that the prior beliefs, the subjective prior beliefs, or the informative prior beliefs can be combined with experimental data, so that the prior beliefs can be updated into our posterior beliefs, which is our belief after seeing the data. And that is Bayesian statistics is based on the Bayes theorem. Just concentrate on the sixth line of the presentation, and please disregard of other mathematical derivations of the theorem. And then the purple parts highlights the version of Bayes theorems that are usually used. And then for application purposes, because of the denominator of the equations in purple doesn't depend on A, so we can put that into the proportionality constant. So the probability of posterior will be proportional to likelihood of the data and then your prior beliefs about the population parameter. So this is an example using a conjugate prior. For example, we have an expert, dermatologist, who is very competent in spot diagnosing conditions. So our parameters of interest is the probability of making a correct diagnosis. But based on our prior knowledge, this expert dermatologist is very, very good. So most of the time he got 19 out of 20 diagnoses correct. So we say that the theta or the prior probability of him making the correct diagnosis is 0.95. And then the probability of him making more than 2 errors is really low. So we can model this prior belief using a beta distributions that has the equations in the red line, in the fifth line. Okay. So after that, we do the experiments. And then when we do the experiment, we collect the data, and then we found out that he correctly diagnosed 9 out of 10 skin conditions. And because of and then the likelihood of such observation will follow a binomial distributions. And the likelihood function is given by the green line. So how can we obtain our posterior belief? So we multiply our posterior with the likelihood function, and then we come up with the results. The one is with the brown line, and then from the equation there, we will know that the posterior distribution has the PDF or the probability distribution of a beta distribution. So we can say that we update our beliefs from beta 0.77.5 with our likelihood, with our data in the likelihood function, and then we obtain our updated belief in the form of posterior distribution of beta 86 and 6. And then we can use these posterior distributions to obtain the 95% Bayesian credible intervals to reflect our uncertainty over the estimates. And then this time, we can safely say that the upper bound and the lower bounds of the credible intervals will will cover 95% probability of obtaining the true population parameter. So this is a counterpart of a base of p-values. So people may ask, I mean, okay, you advocate the use of credible intervals. So how about you want to test hypothesis, and then we have Bayes factor, which is a counterpart of two p-values in the frequentist paradigms, which is just I call that this is just a marginal likelihood, Bayes factor times, which is the probability of 5 given your hypothesis 1 and probability of divided by y with given the probability of H2. Your competing hypothesis. H1 can be your null hypothesis, or H2 can be your alternative hypothesis, or vice versa. So if our alternative hypothesis is H1 and null hypothesis is H2, then the Bayes factors can be interpreted as follows, based on the Cass and Raftery criterion. And if our Bayes factor is more than 100, we can safely say that our alternative hypothesis is the right one, not the null hypothesis. Okay, problems. The problems with Bayesian statistics is the math. The math is pretty difficult, especially if we don't use the conjugate, the non-conjugate prior. We obtain a posterior distribution that has very funny form, mathematical form, that doesn't conform to any standard distribution. For example, in here it's easy because we can see that if we refer to Wikipedia, for example, and then look at the probability distribution functions of the posterior distribution, we can correctly identify it as a beta distribution. But for example, if the likelihood is complicated, we cannot use a binomial likelihood in this case, or we got a posterior distribution that doesn't have any standard form. What we can do? We cannot do. Yes, there's a way. There's a way to circumvent that, but we cannot solve the equation using analytical approach. We have to use a simulation-based approach. Okay. So because of that, we have to, most Bayesian statisticians now, they really have to resort to the Markov chain Monte Carlo procedure, the MCMC for short, which is a computer-intensive simulation-based procedure, and was developed by Alan Galfond and Sir Adrian M.F. Smith in 1990. And because of this, MMCMC has revolutionized the use of Bayesian statistics to the extent that Bayesian data even can be is now being widely used by the non-statistician in fields as diverse as biology, civil engineering, and social science. Two major MCMC algorithms, just for introductions: the Metropolis-Hastings algorithm and deep sampling. The Metropolis-Hastings algorithm, was discovered by a new can be considered as a nuclear scientist, Stanislaw Ulam, during the Manhattan projects, during the development of the atomic bomb during World War II. Then there are other more recent developments. The more approximate method instead of using the computer-intensive MCMC algorithm. For example, using the integrated nested Laplace approximation, using the Laplace techniques. INLA, or the approximate Bayesian computation ABC, or the sequential Monte Carlo method. And these are beyond this talk. I plan to give a talk on this during the weekly seminars in UMBIS as a continuation of this talk. Okay, this is the newest. It's not about the new application, the newest applications of Bayesian statistics in medicine. Can see that the BNT vaccines for the SARS-CoV-19, the analysis of the data was Bayesian. This is Bayesian. as you can see from the results section, that instead of using the 95% confident intervals, the authors or the research teams use a 95% credible intervals to reflect the success rate in preventing a COVID-19. So as a conclusion, Bayesian paradigm can offer an alternative to frequentist methods in analyzing medical research, and then the Bayesian paradigm has become more popular now. The only problem is getting the math right, and then having the right computer or the right procedure or algorithm to compute your posterior distribution. And of that, I see. I thank the audience. Thank you.
Thank you, Dr. Irfan, for your presentation. So finally, we come to our last speaker. Our last speaker of the day is Dr. Lee Wei Kang. Dr. Lee is a senior application scientist from Codon Genomics, who is specialized in molecular biology, genomics, and software technologies. His current focus is to apply biological knowledge and next generation technologies, such as software application, cloud technology, big data analytics, automation, and Internet of Things for precision medicine and agriculture. His topic for today is Big Data Ecosystem for Precision Medicine. Welcome, Dr. Lee.
Thank you for the kind introduction. So hello everyone. I'm Wei Kang from Codon Genomics. So today I'm going to talk on the big data ecosystem for precision medicine, which mainly focus on the data and also the software. So precision medicine is an emerging approach that is just not only tailor the treatment according to an individual or subpopulation genome, but also take into consideration of the environment factor as well as the lifestyle factor, with the final objective to increase the effectiveness of the treatment, to reduce expenses on the ineffective drug, and also at the same time to reduce the adverse side effect from the traditional try and error process in the medical treatment. So the application of precision medicine is directly or indirectly connected to a very broad medical discipline such as pharmacogenetics, genetic testing, epidemiology, and microbiome, and many others that actually generate a massive amount of data. So therefore, the trend in the medical treatment has shifted from the intuition medicine to precision medicine, where all the algorithm and artificial intelligence is used, and also which is heavily depending on the big data. So one of the main type of the big data that involve human genome data, where various countries such as the UK, US, China, and so on has joined the race to sequence and characterize their citizen genome, but other data such as the clinical data, pathogens, environment, and also social data are also equally important. So a very good case study that would be the COVID-19 pandemic, where all these data are actually used together to combat the pandemics. So to manage such a large and also complex data set, so a data ecosystem is very important. So the term data ecosystem is initially used in the business field to describe the enterprise level infrastructure and also application to help the business people to aggregate data, to provide a better understanding on the customer needs, and also lastly to tailor specific sales and also marketing strategy to increase the company profit. So the digital revolution is not only about changing the business field, but also impacting the healthcare sector. So therefore, the concept of a data ecosystem is also applicable in the medical discipline to collect medical data, to enable the medical practitioners to better understand about their patient and also diseases, and finally to tailor a specific treatment that is effective against the disease, which is the precision medicine that we are talking about today. So the big data ecosystem for the precision medicine basically consists of three main parts. So the first part is the data collection, processing, and also the management, followed by the data analytics, and lastly we'll be referring to the integrated platform and also decision support system. So looking at the current trend, there are two main gaps where the first will be referring to the huge data set generated with high velocity, but these data set remain largely analyzed or be interlinked with each other. So another gap that we can actually see currently is the high value decision support system that is still very slow to develop and also implement in the real life situation. So one of the examples could be referring to the COVID-19 pandemic, where we have generated a lot of social and also contact tracing data, but a decision support system that can actually very intelligently process all this big information and combine with other data to give us some informed decision and immediate action is still lacking. So in order to fill in the gap and also to maximize the power of the data, so the digitization and also technologies is very important and is the way moving forward to enable the aggregation of the healthcare big data and also delivery of the informed clinical decisions. So the detailed information of the technologies and how they can actually help to revolutionize the healthcare sector can be found in this MyStick framework released last year by Academy Science Malaysia. So this is a very useful report to see how the new and disruptive technology like advanced material sensor technology, blockchains, neural technology, biosciences technology, and so on, how they will actually play in the role in the healthcare sector in the next 5 to 10 years. So we can actually refer to the Academy Science website to get all this information. And moving into the data ecosystem, in order to exploit the valuable clinical insight from the big data, the very important prerequisite on the data collection, which is to collect a very comprehensive set of the required data, covering all the data so that we can exploit the meaningful information embedded in all this data, with the prerequisite to have accurate data, and also the method to collect the data also need to be very efficient. So this is the summary of the process of the data collection in the healthcare setting, starting from the patient to the diagnostic, health recording, and also biobank, where the real-time flow of the data is very important. We need to gather all this data very fast, real-time, directly from the patients, and follow that will be a high-performing computing to centralize all the data into an integrated database, and also so that we can actually enable clinical decision support tools. So for example, we can actually use all these support system to actually deliver the result to our patients, and also a patient portal. And also for the research wise, we can actually have a integrated database for the information sharing among different researchers from different departments and also discipline. So the centralization of the data is important so that we can actually access the data easily, and also analyze the data from a multi-dimensional view. So, and finally, it will actually come to the patient. It will actually, the new coming trend will be on the patient-generated health data at home. So especially when during the COVID-19 pandemic and also quarantine, so all the digital technologies such as the mobile phone, biosensor, or biometric devices will play a very important role to actually to help the researcher or the medical practitioners to collect data directly from the patient from the home, so that they can actually provide some informed decision on medical advices to all these patients. So this is how the digitization will impact the current medical flow. So in line with this, the use of the traditional method using paper and also the physical file system has to be replaced with the mobile technologies and also the cloud database, so that we can actually overcome some of the challenges and also adverse effects from manual data collection. So for example, we may face problems from the handwriting issue, typo, or even multiple steps of data entry that may create error prompt, those designation, or drug names abbreviation that will actually cause life-threatening consequences in the medical prescription. So this emphasizes the importance of the digitalization to eliminate some of the challenges in the traditional data collections. So for research and also the clinical study, the last data set collected by the researcher must also be sufficiently clean to ensure the data integrity and also accuracy of the analysis. So here I have listed some of the example of the commonly found type of dirty data that is not only applied to the healthcare data, but also other disciplines as well. So conventionally, the data cleaning process has to be performed by an experienced data scientist to prevent the use of the wrong data, to eliminate the structural error that can actually cause mislabel or additional category during the analysis. And finally is to prevent biases that will affect the result and also the conclusion. So the data cleaning is very important. So in the data science cycle, the process of data collection and data cleaning contribute out to 80% of the total time spent by the data scientist. So that will actually indicate how important is to actually perform a professional data cleaning so that whatever medical insight that you get are highly accurate. So therefore, as most of these dirty data are predominantly contributed by manual data collection, digitization and the use of mobile apps could offer a solution to minimize all these biases. So for Quantum Genomics, we are a biological data company that has been dealing with huge data sets over the 10 years of our contract research journey. So by understanding the importance of the data collection, our biologists has worked with our in-house software engineers to develop Darwin apps, which consists of a mobile app part to collect data and a management portal part to store and manage data. So the application of Darwin apps are universal and wide, which could cover from the patient handling registration to biobank management and also finally the result delivery in the healthcare setting. So the key features of the Darwin apps is the flexible form builder where the user of the apps can actually build their own recording easily through the portal interface without the requirement of the coding knowledge. So the resulted form will be seamlessly displayed in your mobile apps and also ready to be used for recording for research or operation purpose. So with the powerful form builder, you can actually use a Darwin app to register a patient who comes to the hospital or a subject involved in your clinical study. You can also register their physical measurement or any type of the research measurement, and also lastly to facilitate the sample collection accurately using QR code. So this is the sample collection using Darwin S is very important to ensure that your sample is will not be missed out or mislabeled and so on. Because sometimes nowadays we also heard of a lot of wrong COVID test result due to the sample being missed out during the process of processing hundred or thousand of sample. So by using digitization, all these limitations can be overcome. So in addition to the function that I mentioned just now, Darwin apps can also be used for inventory management, where the barcoded sample and also barcoded inventory will help the researcher and also the medical operator to track the specific location of your sample with a precise quantity. So you can actually know where your sample is and also how much quantity has been left. And this will be really useful for lab management and also the biobank management. And last but not least, the recording form can be used to capture the test result and can be shared to the patient as a PDF report or to the collaborator as a data set organized in the structured Excel format. So here in this slide, I share some example of our customizable dashboard analytics with the interactive chart for the trial manager to manage the project or for the medical service provider to manage the operation. So all this data you can actually store in the cloud with the centralized data management with some function like editing, filtering, export, and also analysis tool built in to analyze your data. So here is one example of the Darwin application in the human and also the animal trial, where the functions such as Darwin recording, Darwin sampling, Darwin inventory, and also the data management will be applicable in the management of the trial. So if you like to understand more about the Darwin and also the specific case study, you can actually scan this QR code to register your interest, and our team will share more information with you. So overall, the use of the mobile app-based data collection technology, such as the Darwin apps, could help to improve the accuracy and also the reliability of the production data or research finding. So also the standardization and also traceability enabled by the mobile app will help to facilitate future certification and also compliance, especially for the medical sector, and the collaboration among the different departments in the hospital or research institution could be improved so that everyone can access the patient data easily and also to analyze them easily. So lastly, it will also benefit the project manager or the medical operator to provide an overview of the data flow and also the insight that can be gained through the dashboard analytics. So beyond data collection, Cordon Genomics is also providing some data management and also analytics platform, such as our barcoding system to track the species diversity and epidemiology, as well as our genome database to build and visualize genome and transcriptome for research purpose, where some of this platform is being adopted by the leading research institution in Malaysia to serve as their in-house digital platform to support the ever-growing big data research. So in addition to the in-house databases that will enable private data repository, so there are many others publicly available curated database for medicals and also precision medicine, where they are the core component of the overall data ecosystem, and they will require continuous curation from the research community. So one of the examples you can see here is the Cosmic online database with more than 71 million somatically acquired mutation found in the human cancer, where it can actually exploit for precision oncology. So beyond the data management and also database would be the application of the data science to crunch a large data set and also to extract biological meaningful information. There are many machine learning algorithms where some of these algorithm has been mentioned in the Dr. Ifran and also as well as the Chingy presentation early. And all these algorithms are very useful for the future's diagnostic, predictive, and also prescriptive application for precision medicine. So the last part of the data ecosystem for healthcare is the automation of the analytics for real-world application. Because after we collect all the collected data and also analyze them, we need to have a platform that can actually deliver all the informed decision that will actually eventually benefit all the patient and also the healthcare sector. So smart digital health platform will be very important in the future to serve this purpose, especially in the form of the easy to access mobile app, where it will be one of the future focus for Darwin as well for further application in the healthcare applications. So overall, in summary, genomic technology will play a very important role in the precision medicine, at what has been presented by the UMBI and also the Cordon speaker early in this webinar series, especially on the GWAS study and also the ability to sequence and characterize the genetic makeup of the individual tumor for cancer patient. So together with the big data ecosystem involving the software and also the data science, it will actually complete the technical part of the precision medicine. But there are still many challenges exist in the making a precision medicine a great success. But in a positive way, there are many opportunity and also opportunity that we can actually explore. For example, the government initiative in pushing the technology and also providing grant to support the innovation, as well as the increased public awareness about personalized treatment due to the COVID-19 pandemics. So with this, we hope that as a technology company and also Cordon Genomics as a technology and also biological data company specialized in providing a total solution ecosystem involving genomics, software technology, and also big data analytics. We hope to actually collaborate together with UMBI, with all the researchers together, and to create a better future for everyone using knowledge, data, and also technology. So with this, I end my presentation today for the big data ecosystem. Thank you.
Thank you, Dr. Lee. So now I'll open the question and answer session. So again, due to the available internet bandwidth, so I encourage and I would like to remind all participants to type your question on the Q&A feature in Zoom or comment in our Facebook broadcast. So we will compile your question and redirect them to the speakers. So in the question, you please specify your name, the speaker's name whom your question is addressed to, and followed by your question. So I think I already have a list of questions, so not so many. So the first question is addressed to Mr. Subun. So I think it's general audience. So the question is the NGS a kind of genome profiling tool? And what makes NGS different from the microarray? I think, yeah, that's very useful to tell what is the technology feature. Okay, question. Mr. Subun? So the question, I repeat the question. So, the audience asks, is NGS a kind of genomic profiling tool? And what makes NGS different from the microarray?
Sorry. So sorry for the echo just now. I can't hear properly, sir. Sorry. Yeah, actually, NGS is actually can be used for genomic profiling. In fact, it's actually a better, it's offer a better sensitivity at a single nucleotide resolutions. And when you actually comparing both NGS and microarray, they based on different technology in doing the profiling. NGS is based on sequencing technology, where you actually get a sequence of each bases, while microarray based on is a probe-based assay, and you need to actually design the probe or panel to be able to detect, to be able to profile the genomes. So in short, so NGS is actually can be used for genomic profiling, and is often offer a better sensitivity. And it can also be used to uncover rare and also novel genes, because you don't need prior knowledge about the genes. Is the rare novel gene is not required.
Yeah. So
Yeah,
thank
that's
you.
my answer for the questions.
Thank you, Mr. Subun. So I think I have my own question for you as well. I mean, it's writing on the back of this question. So yeah, I mean, in your presentation, actually, you mentioned about the third generation sequencing, right? Can you hear me? You mentioned about the third generation sequencing in your presentation, and it includes the PacBio as well as nanopore sequencing, right? So as far as I know, there are not many institute or university in Malaysia, they have adopted nanopore sequencing. So then my question is nice technology, it's cheap, right? Compared to Illumina or PacBio that cost millions of dollars for the machine. So the nanopore is it's a small gadget, mean ion, it's cheap, and then of course there are different versions. And at the same time also it allows you to do long read sequencing. So what is preventing the Malaysian community from adopting this technology? Because you can bring this gadget anywhere, and then you can do a on-site sequencing, right? So I think what is preventing us from adopting this technology?
I agree that often nanopore, the cost of adoptions of often nanopore is often lower than other kind of platforms such as Illumina. And I think one of the challenges faced by platforms such as ONT is actually they have a lower base call accuracy. Means the raw base call accuracy is slightly lower, and even though it's a long reads, it's a long read sequencing platform side. When you use them to actually assemble large and complex genomes, is often need to be coupled together with short read sequencing technology like Illumina to actually polish it and increase the accuracy of the assemblies. So I think, yeah, and even though the cost of adoptions of the O&D is actually low, but I think the cost of running it, I mean the cost per sample is not as low as when you do when you use technology such as Illumina.
So thank you, Sobun. So I continue with some of the questions. So these questions are actually addressed to Dr. Irfan from UMBI. So the first question is, can autoaggressive integrated moving average, or ARIMA, be used for predicting the values for future, like in the next 30 years?
Yes. For that question, actually, I already prepared the answer for that. I nearly put that on the chat, but I just want to read the answer there. I think the answer for that is both yes and no. Because yes, theoretically, I mean, you can just plug in the values and then get the 30 step ahead of the forecast values in the future using, for example, the ARIMA models. But in practice, the forecast will be very incorrect since there will be lots of uncertainties associated with the errors. In this case, you have to minimize the sum of one step, two step, three steps, and then up to the 30 steps ahead terms. So the forecast value might be way off the correct one. I'm in favor. It will give us a very bad forecast value and the uncertainty around it if the number of step ahead we want to forecast is high. Besides, for the ARIMA model that we use to forecast might be wrong because there will be like changes in the seasonal trend and the cyclic behavior of the time series data set. So again, the forecast value might be wrong. I'm in favor that it's not advisable to forecast like a 30, of doing like 30 step ahead and obtaining the forecast value like for 30 years after the last observation in your time series data because of the error associated with that, I think. Yes, that's my answer. I will post that on the chat.
Yeah. And the second question for Dr. Irfan, do we always follow the 70-30 rules for prediction and forecasting? What kind of scenario when we do not use the 70-30 rules? And if so, what ratio will you reckon?
Oh, for this one, I have to say that I'm a bit unfamiliar about the 70 and 30 rules for the prediction. So for that, I have to pass to anybody who knows the answer for that. Yeah.
Yeah, maybe. So in what circumstances do you think the Bayesian theorem should be applied in biomedical research? So that's the last question for you.
Okay. In my medical research, I always give this example. For example, that we want to do like GWAS studies. GWAS studies involves lots of, SNPs, can be like how many? Like 1 million SNPs that you want to investigate for certain conditions. And because of this, in the frequency settings, we sometimes use the lasso techniques for that. I mean, we put a L1 or L2 penalty term in your regression model, and after that we can shrink any unnecessary regression coefficients to zeros, so we can reduce the dimensions of your regression models. I mean, from something to something lower in terms of lower dimensions, and because of that, you can stabilize the regression models more. But in all the SNPs, some of the SNPs, we might have a prior information behind that. And when we just use the SNPs just to control or to adjust the models, I mean, because of that can be the source of confoundings for the models. Because we have that some prior information about that, we can use a prior. Because I call that a spike and slap prior to reflect your prior beliefs about the regression coefficients. We can put our prior belief on certain SNPs as zeros, and then certain SNPs might be one. I mean, depends on the expertise that we have at the time. It depends on the level of knowledge we have at the time, and then we run the models using a lasso regressions or elastic net regression if we want to include both the L1 and L2 penalty term. And then we can get a posterior estimate. And then because of that, the shrinkage posterior will be much better in terms of reducing the dimensions of your data set, which is good.
So, thank you very much, Dr. Irfan. Actually, when I checked my Facebook page, actually, Dr. Shakima also has given a very, very excellent explanation on the difference between NGS and also microarray. So I think it's important. I think if we encourage everybody to follow our Facebook page, actually, so I can see the answer is very long and then very comprehensive. So yeah, I think I have got another new question from Dr. Wan Muhammad Aizat from InBiosis. Yeah, he thanks Umbi and Cordon Genomics for the interesting talks. And then he was wondering, and what is the current status and way forward for the Malaysia cohort? And are we looking at any potential drugs, treatments in the pipeline to specifically for Malaysian population? So this is a question from Wan Aizat from InBiosis. So anyone from the Malaysia cohort can answer this question, maybe.
Thank you, Dr. Tek Yu. For that question, yes, we are doing all the genotyping analysis to way forward for precision medicine in Malaysian population. But as you can see from our results that we presented here, the results show that genotyping results show a very small effects, which is less than 2% for each population. So for clinical, we are here we can see although it is significant, statistical significant, but for clinically significant, it is too small for us to translate it into clinical practice. So maybe as you can see in the presentation just now. So here now we have a current method that can overcome the small effects analysis that we are facing previously. So we hope that in the near future that we can do more analysis for the small effect genotyping analysis that can we use to translate it for the clinical practice. Thank you, Dr. Tek Yu.
Thank
I
you.
hope that will answer.
Yeah. So I'm looking at the question that is coming in. Maybe we could wait a while. And yeah, I hope everybody, I encourage everyone to ask questions. So yeah, before that, I think I have a question for Dr. Shakima because in Dr. Shakima's slide, actually, she presented the LRGF,
Yes.
right? Okay. So among the patient in Malaysia, actually you have the hyper mutated and the so actually one patient with the MSS, what do you call that? The microsatellite stable patient actually has a lot of mutation. It's hyper mutated. So since it doesn't have a MMR, so what could be underlying the hyper mutation in this MSS patient?
Okay, thank you, Professor Madya, Dr. Lau Tek Yu. Interesting question. So those who really focus on my presentation will notice that one of the microsatellite stable patients among the colorectal cancer patients actually has hypermutation. Okay, firstly, I have to mention here that the protocol that we use for detecting the microsatellite instability is the Promega assay kit, whereby the markers included inside the Promega kit was actually derived from other population, which is the Caucasian population. From my discussion with the collaborator yesterday, when the panel for macrosatellite instability testing was derived from a group of person that has kind of different genetic makeup from Malaysia population, the rate of microsatellite instability detection is low. And that is what was discovered from our study. So the underlying mechanism is that there could be other type of genes that could contribute to hypermutation phenotype. And not only those MMR gene, but it could be other genes, whether the genes are upstream or downstream of the genes that we are studying, but those genes are not included in the Promega panel.
Yeah, I get it.
Those, yes, and those gene, the alteration that lead to hypermutation could be due to very large variation in the copy numbers, or it could be due to large deletion in certain genes that is contributing to mismatch repair.
Yep. Thank you, Dr. Shakima. So yeah, I think we still open for questions. So I mean, maybe the question can come from the panelists as well. If you have any questions. Yes, I think for codon, for data scientists from codons, right? So I think I mean it has been the last 5 years has been amazing, right? Because I would like to get your opinion as a data scientist, right? Because the last 5 years I was trying to learn big data. Then within that 5 years, like then you have AI coming out. Then after AI, then blockchain, and then now we have like quantum computing, right? Quantum supremacy. So then so there's a lot of new computational tools that's coming out. So uh, so how would that affect the business of, let's say, DNA sequencing and things like that? So I'm not sure. So do you have anything to share with us?
Okay, thank you. Okay, so thank you, Dr. Thank you for the question. So based on the question, so some of the challenges that we can see from the a lot of technology, like what you mentioned, the blockchain and all this technology actually generate a lot of data. So when we deal with a project and also with the industry and all that, first, one of the challenges is that the data is too big, and also there are a lot of unclean data, like what I mentioned just now. There are a lot of dirty data and all that. So one of the challenges is the ability to actually to clean all this data, and then followed by the next one is the how it can actually industry process all the data together. So we always face challenges and problem from the client that there are too many different type of data, not only the genomic, but also the social data, the physical data and so on, how we can actually put all this data together. So it's very different from the conventional research where we have only a few variables only. So when all these are a lot of variables and also parameter come together, then you will actually create the needs for the data science and also the large scale data analytics to actually to help them to interpret the data. So they need a data exploration like what Qingyi mentioned, to provide a helicopter view of all the data and see how all this data being connected together to give them a big picture. So these are some of the challenges that we observe for the past 2 or 3 years, and also I will actually see this will also the coming challenges in the next 3 or 4 years. Definitely, it's easy to adopt the technology, the latest biosensor or the sensor technology, but when it comes to the data generation part, there are still a lot of limitation and bottleneck where will require data science and software to fill in the gap.
Yep.
I hope this answers your questions.
Yeah, thank you, Dr. Lee. Yeah, I think it gives me a lot of information about this new technology, because my time is still with the data science, so but it has advanced so much. I mean, all the computational tools, and yeah, I think I agree with you that that is what also Ms. Cheng Li is trying to do, right? So she's using machine learning to identify between the benign and the malignant breast cancer tumor, right? And then you try to discriminate between the benign and malignant tumor type based on the clinical feature of the cancer, right? So in this case, yeah. So in this case, but you are not using the raw data per se from let's say the what do you call it, the ultrasound or whatever scan, right? You're just collecting the data from the Wisconsin data set itself.
Hi. So yeah, I'm not using the raw data. It's a online data set. So I think is they use some software to process the raw data, and then they will get all the variables that I show in my presentation. So there is actually have some nowadays there's a lot of imaging analysis. There's a lot of statistical or computational method to do analyze this image. So all this algorithm is very advanced. So it's actually quite accurate now. Like especially we can use the deep learning to analyze those image. So we will get quite a reasonable result and accuracy.
And this is also one of the direction of codon genomics as well, because I read somewhere that uh, it may make the pathologist lose their job because the algorithm is better in diagnosing the cancer or the hemorrhage from the patient compared to the doctor or the pathologist themselves to identify the lesion. Yeah.
Yeah, definitely. Yeah, this is a good direction, and then there's is so we can use this model to predict the anything you I mean any predictive model to help you to have a more subjective decisions. Yeah, and make you let's say more efficient and also yeah.
Yeah. Thank you, Miss Chingy. So actually, I received a question. A lot of people actually are interested to get the slides, or they want to watch the replay of this session. So yeah, I think for those who are really interested in our, a replay of the session or look at the slides, maybe one good advice is for them to come back to the UMBI website, because we will keep the video, and then you can just follow our social profile, and then you can get access to the video as many times as you like to. So that is no problem. Yeah, in, that's all. I think we do. Do you have any more questions? It's almost 5 o'clock also, right? So yeah. So yeah. So since it's closing, I mean it's closer to 5 o'clock. So I think finally all good things must come to an end. But before we end this seminar, we would like to thank all our participants for actively taking part in our very meaningful activities. We would also like to thank all our honorable speakers as well as the organizers, the management of WUMBI and Codon Genomics. And not to forget our very hardworking and also very well-trained IT support team to make this thing happen, right? So WUMBI has a very long history collaborating with the industry partners, and especially Codon Genomics. So we hope this webinar can be a stepping stone which further renew and cement our existing close ties, and to promote exchange of ideas and expertise between academia and the industry in a win-win manner. And since, as Dr. Lee mentioned, the research scene in Malaysia is getting more and more translational, and also a lot of grants are open to both industry and academia. So I think it's a good thing that we plan to host more and more such industry and academia collaborative webinar in the near future. So in addition, with this webinar, we also hope that we can provide a useful platform to help our scientists to keep the science going at this depressing moment of the pandemic, right? So one last thing. So if you find our webinar useful and interesting, and if you would like to join us and learn about what we do, or simply wish to get a glimpse of our future webinars, We seriously encourage you all to follow our websites and social media profiles. And finally, for attendance and Easter purpose, please click on the links provided at the end of this session. And yeah, you can follow both UMBI and Codon Genomics website, Twitter account, as well as Facebook at the following link. And everyone, please take care and we'll see you again. Thank you. Thank you very much, everyone. Bye-bye.